And we're very lucky to have with us today Justin Carré from Nipissing University, an esteemed colleague in the area of social endocrinology. We almost lost Justin from science to hockey, I think, at the outset of his career, played junior A hockey, and went to Canisius College on a NCAA hockey scholarship, right? Early on, where he did his undergraduate and master's work, and then saw the light, being neuroscience, and moved to Brock University to work with Cheryl McCormick at Brock. And while they were working on the PhD, launched a really impressive research program and started racking up publications. Notice right from the start, I think I must have heard about you almost the day it started in that program, it feels like. He did a postdoc at Duke and was recruited to Wayne State in Detroit for a couple of years before Canada managed to seduce him back to go to Nipissing University in North Bay, where he's built a large and impressive research operation and is now a full professor. Because going through your CD, there's so much in there. One of the things that was striking is that in the time he's been there, Justin's published more than 100 papers. He's had many great grants, and is, I think, the only person I know who has simultaneous operating funding from NSERC and SSHRC, which is kind of amazing. So it was cool to see that. So a number of years ago, I had the good fortune to connect with Justin through a couple of particularly gregarious grad students, and he kindly invited me to collaborate with him on his work looking at behavioral consequences of administration of testosterone to healthy participants in a very large cohort. I think it was, and maybe still is, the largest study of its kind in the world.
It seems like we got to get more and more people all the time because our statistical models are getting a bit more sophisticated.
More complicated. So I'll be very excited to hear the latest updates from Justin's lab and from that work. He's going to tell us about the use of single dose administration of testosterone in the role of testosterone in modulating human aggression and mating psychology. Justin, welcome.
Well, thanks, Neil, and thanks for the very nice, kind introduction. Now I feel I'm going to let everyone down this talk and tell you all about the research. I'm going to just start this by saying it's going to seem a bit like a job talk because not that I'm looking for another job, but it's going to be sort of a here's where we started and kind of here's where we are right now, and talking about this, how the studies each build on each other. And one of the things that some people find boring, I find important, is the use of replication, and not just getting one finding and then jump into the next thing. And, so we've sort of built in these replication extension in a lot of our studies. So, make sure we're checking ourselves and making sure that some of these findings are not just one-off. So some of it might seem a little bit repetitive. We add a little bit of, we always try to extend it in some important way, but there's some replications built into here. So those of you who don't know, Nipissing University is a really small liberal arts school in Northern Ontario. We have, I think it's about 6,000 students, so it's pretty small. When I left Wayne State University in Detroit, Michigan, after 2 years on a tenure track there, my colleagues thought I was nuts. They're like, "What do you want to do at Nipissing? You're not going to be able to continue doing all this cool research on testosterone and behavior." I said, "Well, that might be the case, but my wife and I are both from Northern Ontario, and so we want to move back home and be close to family." And I've been fortunate enough to get, NSERC, CFI, not CHR yet, but CFI, and other grants to be able to continue to do this kind of research. So so let's just jump right into it. So one of the cool things that what is going on here? One of the really cool findings when I was a master's student and a PhD student, at least for me, being a competitive athlete, was the finding that testosterone levels respond very rapidly to competitive interactions. And what's even more cool, I think, is that this pattern of findings is similar across many different species. So you see it in rodents, you see it in fish, you see it in birds, non-human primates, lizards, which together suggests, and even humans, these are my 3 kids that are always showing off their trophies and medals and so on. But which is, I think, is cool that when you can find a neuroendocrine mechanism that seems to respond to competitive interactions across many different species. I think it means that hormone change is potentially very important for regulating ongoing and or subsequent behavior. So there are many theoretical models over the years. The challenge hypothesis by John Wingfield suggested that testosterone levels fluctuate throughout the season in bird species, being at a very low baseline, rising to a level B, which he calls breeding baseline to support reproductive physiology and behavior, and then further rising to physiological maximum, which they posited was to support aggressive and territorial behavior. Alan Mazur came along later and showed that these changes in testosterone seem to depend on the outcome of the competition, rising in winners relative to losers. And his model would suggest that an increase in testosterone among winners might promote competitive behaviors, aggressive behaviors aimed at maintaining high status, whereas a decrease in testosterone among losers may promote more submissive behaviors aimed at avoiding further threats to status and physical injury. So my research as a PhD student was focused primarily on that second component, whether these acute changes in testosterone have any impact in people, and in particular in predicting future behavior. And so how do we go about doing that? And sorry, this is John Archer's. This is right around the time where I was proposing my PhD. The other side of his hypothesis, and the whole point adaptive terms, the surge in testosterone should increase aggressiveness in competitive situations. So in 2006, when I was proposing my PhD, this was a question that nobody had really addressed in human studies. So how do we go about testing this? Well, pretty simple. You can have people engage, and I say it's simple. It's a little hard. When I was at Wayne State, I had to purchase Xbox video game and TV and so on, and my administrators said, you can't buy video games. Like this is not how a tenure track faculty member does. And no, it's part of the research. And so anyway, so we can rig a competitive interaction such that we have participants play against a very easy opponent or play against a very hard, difficult opponent. And you can track changes in testosterone that occur in saliva. And for those of you who are not familiar with this, saliva is a pretty decent estimate of what's circulating in blood. So if you collect saliva in blood and you look at the correlations, they're very strong. So you can measure change in testosterone in response to a competitive interaction. You can see how it differs between winners and losers. And then you can assess later on, once you've tracked that change in hormone, you can assess a behavioral outcome. We've typically focused on aggression. So we have a task that I'll describe a little later that is a pretty good estimate or a pretty good proxy for measuring reactive aggression in a lab-based setting. And this is just one example of what we found in the past, so that a rise in testosterone positively predicts subsequent aggressive behavior. We find it in both winners and in losers. What I'm not showing you here is females. So this is a study, we had males and females, and in this study we found the effect in males and not in females. And so there could be a variety of reasons why the effect wasn't found in women. One part is that it's really hard to measure low concentrations of testosterone in saliva in women. So measurement error may in part explain why we don't find these effects in women. And when we look at a meta-analysis that looks at whether basal T maps onto aggression, change in T maps onto aggression, or exogenous T maps onto aggression, what we find is the strongest evidence is for change in T in response to a competitive interaction. This seems to be the strongest predictor of subsequent aggressive behavior, at least in men and not in women, as you see here. So this is a 2020 papers. Since then, we've published several papers with exogenous administration. We haven't yet revise this meta-analysis, but it seems to be that exogenous T can have a relatively weak but significant effect on the aggressive. Okay, so the problem here is that even though we're able to separate in time the change in hormone and then measure subsequent behavior, so we know that it's not behavior that's pushing a rise in testosterone, but rather it seems to be a rise in testosterone that might be pushing behavior. We have a potential third variable problem, whereby some unmeasured variable is both increasing testosterone response to competition as well as making one more aggressive. So what we might be seeing is just an artifact. So to really get at this, we have to develop pharmacological challenge interventions where we administer testosterone or a placebo and we see how it impacts aggressive behavior. So one of the first studies that we did when I was at Nipissing University, we collected data from 120 healthy young men. We randomly assigned them to receive either testosterone. In this case here, we used a transdermal gel placed on the upper shoulders. It's a gel that's used typically to treat hypogonadal men, or they received placebo, so between subjects design. And I don't have time to get into the why for this as much as just trust me that these individual difference variables are important. So we looked at personality traits that might influence the degree to which testosterone predicts subsequent aggressive behaviors. So we looked at trait dominance, we looked at trait self-control, and we looked at self-instrual, which some of you may not be familiar with this. This is the extent to which you consider yourself as relatively independent of others or connected to others. So higher score means you're more independent in your orientation. And the reason we looked at these variables is that our correlation studies suggested that changes in testosterone, natural changes in testosterone, interact with these personality traits to predict aggressive behavior. So we included them. Then this is just our basic design. So in our earlier studies with a smaller sample size, we were able to do blood draws, which is the best approach, but it's also invasive and costs a lot of money with getting a nurse practitioner to do blood draws. But it's a basic design where we collect consent, we get their self-report personality measures, we get blood samples, they get a resting period, and that resting period is 60 minutes. So we had done a pharmacokinetic study before this study to show that it takes about an hour for androgel to placed on someone's shoulders to increase testosterone levels to high normal range. So we waited 60 minutes, and then we had them play this game called the point subtraction aggression paradigm, which I'll describe to you in just a moment. So the PSAP was taken from Don Cherrick, who developed this in the 1980s to study the effects of different substances like SSRIs or alcohol administration and how it might impact aggressive behavior. And the way this task works, and I'm going to show you a little video of my son playing it when he was really little. But the way this task works is you're bar pressing for points. So you press that button number 1 100 consecutive times. What happens is your point counter at the top, at number 12, will start flashing with positive signs around it, and it'll increase by 1 point. If you continue to do this, you get more and more points, and we tell participants the more points that you earn on this task, the more money you can pay at the end. So these are undergrad students that are looking for money, and so they are working really, really hard. We've had to replace many keyboards throughout the years because they're just hitting buttons very hard. Now, what we tell them is we're pairing you with another person who's in another room, either here or another university, and they're playing the same games. And they're connected, your computers are connected, and it could occur, it may occur depending on your partner's strategy, that your point counter may start flashing in red with negative points around it and decrease by 1 point. If this happens, it means that the other guy stole a point from you. The other guy gets to keep all the points he steals from you. Okay? We tell participants you can respond by continuing to hit that reward option, button 1, or you can revert to button 2 or button 3. If you hit button 2 10 times, what's going to happen is you're going to steal a point from the person you're paired with. However, we randomly assign you the condition in which you do not get to keep those points. Okay, so the other guy's going to lose them, but you don't get to keep them for yourself. And so the reason we introduce that rule is so that we know that participants are stealing points from the other person in order to get back at them, not to gain any financial reward for themselves. So it's a behavioral assay for reactive aggression. And then they have a third option, which is button number 3. If they hit that 10 consecutive times, we tell them it'll protect their point counters against subtractions for a variable amount of time. Okay. In reality, they're not playing with anyone. It's just a bot that's designed in a way to provoke you throughout the task. If you hit button number 2 or button number 3, it'll initiate a propagation-free interval, which means that nobody's going to steal from you for about 45 seconds, at which point random subtractions will happen again, so that it sort of mimics a real type of interaction. So this is what kind of looks like. So this is my son. I think at the time he must have been like 6 years old. He's 13 now. I told him he was playing with another kid his age and give him the same instructions I just gave you. So let's just show you this quickly here. So you get his point. So I'm just pausing that for a moment because what I didn't tell you is that once you commit to an option, you're stuck with that option until you finish this fixed ratio. So if you're at like button number, you've hit button number 1 22 times, you can see there he was trying to steal back right away. Like he was like trying to steal back, he wasn't able to, and he remembered the instructions that once you hit it once, you got to hit it 100 times. So now he's going to go back to hit it number 1 for 100 times.
It's like he's using facial expressions, that's an 80.
So what I want to point out here is that, and we'll get to this at another one of our studies, but one of the we're trying to understand mechanisms as well as to which testosterone increases aggression. And one mechanism might be through anger, it makes you more angry in response to provocation. Another mechanism might be through reward value, so it feels good to steal back. And I think that arguably you can see his face when he stole back, he's like, yeah, like I got him, like you know. And so it seems, at least from this video, one may argue that it might be through a reward mechanism. So that's how the task works. Now just jumping into the results. So this is just showing you the pharmacokinetics. Just a reminder, this is 60 men who got testosterone, 60 got placebo. And what we're showing you here is changes in testosterone over time in response to your treatment. Here we have testosterone group clearly shows an increase within 60 minutes, and it's sustained throughout the experimental paradigm. Placebo group, no change. The one important point I want to make here is we're pushing them to the high normal range for men. Okay, so men range from about 3 to 10 nanograms per mil. We're not pushing them above the normal physiological range. We're mimicking what would happen in a regular type competition. So about a 60 to 70% increase in testosterone. So then our behavioral data, so this is aggression, and what we find is that the testosterone group is 27% more aggressive. From a statistical significance standpoint, it was just short of significance, but I think it's important to just show you that the main effect was such that the testosterone group tended to be more aggressive on this task. Now, jumping into the individual difference variables, so as mentioned, we measured trait dominance, trait self-control, and trait self-control. And for all three of these individual difference variables, it seems to be, they all seem to have an impact on whether testosterone increases aggression. And so here's one graph, and basically what this is showing you is a significant trait dominance by drug interaction. And when we do simple slope follow-ups, we find that testosterone increases aggressive behavior, the orange line, but only to the extent that you score high on trait dominance. Okay, so for guys who score low on trait dominance, testosterone has zero impact on aggressive behavior. When we look at trait self-control, find something similar, although it's the other direction because the effect is only found in men who score low in self-control. Okay, so testosterone increases aggressive behavior, but only among men scoring low on self-control. And for self-control, the interaction wasn't quite significant, but it was in the predicted direction such that testosterone increases aggressive behavior, but only to the extent that you score more independent in your self-control. Self-control is the extent to which you consider yourself as independent of others versus interconnected with others. So you can think about like individualistic versus collectivistic orientations. And so this maps on pretty well too, like in North American society, people are higher on independent self-control, whereas somewhere like China, people are higher on interdependent self-control. So what we've done too, and this is in part based on reviewer input. So sometimes, a lot of times, I don't know, reviewers always provide or sometimes provide really good input to how to potentially improve your paper. One of the concerns is that when we have these three individual difference measures, of course, we're going to find something that works. You throw enough crap on the wall, something's going to moderate the relationship. So they suggested that we do like some of the work in behavioral medicine or instead of behavioral medicine, but some research looking at one's likelihood of getting a heart attack, for example, and that would be best predicted by a composite risk score that takes into account many factors like cholesterol levels, blood pressure, heart rate, socioeconomic status, et cetera, et cetera. So what we did here is created a personality risk index. So we standardized each individual difference measure, and then we took the average score to get at a risk index that might better encompass all three of these moderator variables. And not surprisingly, what we find is people who score higher on this risk index, testosterone has a strong potentiating effect on aggressive behavior, whereas those scoring low on this personality risk index, testosterone has zero impact on aggressive behavior. So, so that was exciting. And this is when we looped in Neil, and I believe Tanya was one of your students, right? So one of Neil's students in this project. So we were interested in replicating and extending this work. The replication involved a few things. We used a different drug, so we used intranasal testosterone. The reason we chose intranasal testosterone is it acts a lot quicker. So within 15 minutes, we see a substantial increase. It's also not as messy, so my research assistants would have to apply the gel on men's shoulders. It would take an hour to anyways, intranasal testosterone seemed much better to use. We also include our individual difference measures, calculated personality risk. We upped our sample to 400 men. And we looked at genetic variability in the CAG repeats of the androgen receptor to see whether this effect that we're finding is likely through an androgen receptor mechanism versus potentially an estrogen receptor mechanism. And finally, the other thing we added here is after the point subtraction task, we asked participants 2 questions. We asked them, if your partner, to what extent did it make you angry when your game partner stole points from you? And we also asked them, to what extent did it make you feel good to steal back from your game partner? Okay, so we're trying to get at one potential psychological mechanisms which testosterone modulates aggression. So this is Neil. I think that Neil, this might be an older picture. Maybe not. And so we looped in SFU colleagues to help us with genetic analyses, and Tanya came and spent some time at Nipissing University to extract DNA for us from, I believe it was mouthwash samples at the time. And so we were able to quantify the number of CAG repeats within the androgen receptor gene. And the number of CAG repeats is inversely related to the transcription efficiency of the androgen receptor. So the fewer CAG repeats you have within exon 1 of the androgen receptor gene is associated with greater transcriptional efficiency. So we expected, or based on this, we expected to find that our effects of testosterone on aggressive behavior would be most strong amongst men with fewer CAG repeats within this androgen receptor gene. Some in vitro research as well as in vivo research supports this. So to the left here, you see they created androgen receptor protein that had 15 CAG repeats, 24 CAG repeats, 31 CAG repeats, dosed them with an androgen, in this case dihydrotestosterone, and found that the best transcriptional efficiency was with the fewer CAG repeats relative to more CAG repeats. On the right, you see these dark circles are those who got testosterone, and the light circles are those who got placebo. And this is looking at changes in prostate volume. And what you find is there's an increase in prostate volume after testosterone administration, but only among those men who have fewer CAG repeats, not among those who have more CAG repeats, which again suggests that having fewer CAG repeats, your effects of testosterone might be more pronounced. So this is our pharmacokinetic study. We did it with a smaller sample size of 13 men, repeated measures, crossover design, just to confirm that our manipulation would increase T. And again, we have them within 15 minutes, you get a substantial rise in testosterone in the high normal range among those who got the 11 milligrams intranasal gel, and no change in testosterone in the placebo group. So what do we find? Again, we find evidence that testosterone does seem to have a small effect on aggression. This one here also just failed to reach statistical significance as a main effect. But what we find is, again, this exact same interaction we reported in our previous paper. So personality risk index moderates the effect of testosterone on aggression, such that testosterone potentiates aggression, but only in men who score high in this personality risk index, not in men who score low. So as I said earlier, many people will find this boring. It's just a replication of your previous work. But I think it's an important one because we at least have more confidence that that first effect was not just a fluke. So here we extend this finding by demonstrating not only a risk index by drug interaction, but there's a risk index by drug by CAG repeat polymorphism interaction. So what we find is that for those men who have fewer CAG repeats or a more sensitive androgen receptor, we find that this effect is quite robust, so that testosterone increases aggression amongst those with who score high on the risk index, but not low on the risk index. When we look at guys who have a we'll call it less functional androgen receptor, you have absolutely no effect. So testosterone has no main effect, and testosterone does not interact with that personality risk index to predict aggressive behavior. So this suggests to us, although it's, the CAG repeat, as much as it's a sort of genetic marker, it's still correlational. So we didn't, manipulate the availability of the androgen receptor using flutamide, for example. But it certainly does suggest that our effect of testosterone is probably through an androgen-related mechanism, not an estrogen related mechanism. And I should say why I'm talking about estrogen. I'm not sure how many people know this in the room, but testosterone can get converted to estradiol via an enzyme called aromatase. And so some people, animal researchers, at least in rodents, many studies in rodents and birds, find that effects of testosterone on behavioral outcomes seem to be mediated via an estrogen receptor mechanism. So testosterone gets converted to estrogen via aromatase. And so but for the next NSERC that I apply to next year, I think we're going to try to add a aromatase inhibitor to our manipulations to more really nail that down. Okay, so then the next question is, does the effect of testosterone on aggression in guys who have high risk personality indices and low CG repeats, is this effect mediated through anger in response to provocation? Or is this effect mediated through this feel-good reward-related mechanism? And so what we find is, long story short, doesn't seem to be anger. It seems to be reward. And so essentially, when you control for the extent to which they feel good, this led to a 78% reduction in the magnitude of the effect of testosterone on aggressive behavior amongst these men who score high on that personality risk and low in CG. So it seems to be a reward-related mechanism at play. Okay, so now I'm going to sort of jump into, just going to try to keep track of time here. I'm going to jump into a bit of a different area of research that I got involved with, and I think many of you know this as our faculty and those who are following online. Your students take you in directions that you weren't necessarily anticipating going into, and this project here was actually a course I taught, a hormones behavior undergraduate course that I taught. And the students were responsible for writing a research grant, like a really short research grant. And she had proposed looking at the effects of testosterone on mating-related psychology and behavior in people. And it was based on an animal study that demonstrated that testosterone and I hope I remember this here correctly. I think it was California mice. Testosterone administration to California mice increased mating effort, and I think it was ultrasonic vocalization that looked, but increased mating effort for unpaired California mice, but it actually inhibited ultrasonic vocalizations for paired California mice. And so she was interested in the extent to which the effects of testosterone on mating psychology would depend on whether you were paired or whether you were single, with the prediction that testosterone would upregulate one's in this case here, we use ratings of attractiveness, would upregulate ratings of attractiveness for single guys, but would downregulate ratings of attractiveness for paired guys. So this is a task, so I won't bore you too much about all the details, but we use intranasal testosterone again. This is 120 men within subject crossover design. So on one day they got testosterone, on another day they got placebo. Of course, it was double blind counterbalance. And one of the tasks you can see at the bottom is facial attractiveness ratings. And so the girl on the left, these are standardized images that are publicly available. And not only are the images available, but the ratings of thousands of men who rated them on attractiveness are also available. So we're able to select faces that were rated low on attractiveness, which in this case was the picture on the left, or high on attractiveness, picture to the right. And so we call that consensus attractiveness. So your men are rating these faces of women that were previously rated as low or high on attractiveness. Okay, so what did we find? Well, we found a complex three-way interaction. So a drug by relationship status by consensus attractiveness. So on the left, I'm going to show you first the ratings of the images of women who were previously rated low on attractiveness. Okay, and what we find is that for single guys, you see the ratings under placebo are quite low, but they increase as the ratings run of a 5-point scale. But testosterone upregulates the ratings of attractiveness from these relatively unattractive female faces. Okay, and if we look at partnered men, testosterone does nothing. So testosterone has no impact on ratings of attractiveness from relatively low attractive female faces. And when we look at women who are high attractive stimulus images, we find that single guys, regardless of whether they get testosterone or placebo, they rate them as high in attractiveness. Okay? So testosterone doesn't seem to have any impact on attractiveness ratings for high attractive faces for single men. But for partnered men, they show a bit of an upregulation. And so one other thing I want to point out is these partnered men on placebo, they rate these high attractive faces as kind of low on attractiveness. And this has been thought of as being a sort of relationship mechanism whereby men simply underrate attractive females as a way to, keep the relationship in good standing type of thing. So, so basically what we find is that testosterone increases ratings of attractiveness for single men when they're rating low attractive faces. And it tends to increase ratings of attractiveness for paired men, but only if these are highly attractive faces from the stimulated views. Okay, so now we're going to go to another student of mine was interested in this psychological construct of dehumanization, and this is rating people lower on uniquely human traits or emotions. So again, we had 120 men within subject crossover design, and these men were randomly assigned to watch a video of one of two Taylor. This is our actor that we hired, and Taylor dressed in one of two ways. On the left, she dressed with a Under Armour hoodie. Her hair tied up, no makeup, sort of more of a traditional like student on campus thing. And this is the same person on the right, Taylor on the right, dressed in a more revealing way with a low cut shirt, her hair done, makeup on. So we call these a conservatively dressed woman on the left and a more sexualized dressed woman on the right. So men would watch a video of Taylor describing herself, and both videos were the exact same. So they were just saying, my favorite sport is this, I like country music, my favorite artist is this person, and so on. It was a 3-minute video. And after watching that 3-minute video, they rated her on a number of what are called primary and secondary emotions. So primary emotions, also known as non-uniquely human emotions, these are things like excitement, joy, pleasure, fear, pain, rage. So these are emotions that are typically experienced by all animals. And secondary emotions are thought to be more uniquely human emotions. And these are things like compassion, hope, guilt, remorse, despair. So they rated Taylor on all of these different emotions, and we can calculate a measure of dehumanization by taking primary emotions minus secondary emotions. And so the higher you score on this index, the more you are dehumanizing the person within the video. So what did we find? Well, we found a significant drug by type of emotion, by type of stimulus interaction. And so what we found, I'll show you the sexy condition. What we find is evidence for dehumanization regardless of whether you get testosterone or placebo. So men, whether they get testosterone or placebo, will dehumanize Taylor, who's dressed in a more sexualized way. And so that is, they score higher on, they rate her higher on primary relative to secondary emotions. And if we look at the conservatively dressed condition, what we find is that for the placebo condition, so in red, you have no evidence for dehumanization. So they rate them similarly on primary and secondary emotions. But what you have is testosterone leads to a dehumanization of the conservatively dressed female stimulus. And so, so that was, I think, one of the first study to demonstrate this effect of testosterone, and it seems to depend critically upon the type of outfit or presentation of our stimulus that we're using. Okay, now we got I had a PhD student when I was at Wayne State University. His name is Stefan Getz, and Stefan was, when I left Wayne State University, he was in the middle of this project, and unfortunately wasn't able to come to Nipissing with me because we don't have a PhD program in psychology. So he continued on with his PhD project at Wayne State University, and one of his interests was in looking at the effects of testosterone on mating type behavior in people. So when I got to Nipissing, I said, well, we can collect these data for you and send you the data, and you can write it up. So this was his project. It was part of a larger project that a collaboration with colleagues at Stockholm School of Economics in Sweden. And they wanted to look at the effects of testosterone on economic decision-making in a very large sample. So we collected data on 1,000 healthy young men over the last 6 years at Nipissing. 500 got testosterone, 500 got placebo. Now, after that study was done, so it was a 2-hour protocol, we invited participants to participate in another short study on, we called it person perception. So fast friends kind of interaction, person perception. So this is basically our setup. This is in a conference room. The female on the left is a paid Confederate. She's actually an actor in North Bay. We have a pretty vibrant movie scene in North Bay of all places. I think Vancouver has one as well. And so we hired her, and her job was just to sit there across from the participant who arrives and to not say anything for the first minute. And we were looking at latency to speak. So is testosterone going to increase men's latency or decrease their latency to speak to the woman? Is it going to make men, speak more about themselves, smile, et cetera, et cetera? And not only that, but we had people code for her behavior and for his behavior. And one of the reasons for that is probably within the first week, our female actor approached me and said, I know I'm supposed to act the same with every male, and I'm trying my best, but it's not possible. Some men that come in here are very attractive. Some men who come in here are very unattractive. And I have a feeling that my behavior is not the same across these, conditions. So even though we had a professional actor, she was, knew enough that she felt that she was not able to act the exact same depending on attractiveness of the male. So we were able to code her behavior for based on previous research, and this behavior was we call them 9 affiliative behaviors, looking at things like how much interest did they show in the person, how talkative was she, how much was she smiling, etc. And these 2 trained raters were blind to experimental condition, and they were also blind to study hypotheses. So we were able to calculate or code for the Confederate's affiliative behavior. And the other thing is, when that 3-minute interaction was over, we took the male participant to another room, gave him an iPad, and had him fill out questions. And some of these questions were, how attractive do you find this the woman you just interacted with? Another question was, to what extent do you think this woman would like to have a one-night stand with you? To what extent do you think this woman would like to have you as a long-term partner? And we asked a few other questions like that. But my student was mostly interested in short-term versus long-term. My interest as a short-term partner, long-term partner. Ended up being that these things were so highly correlated that he combined them into a single index of interest in, or his perception of her interest in. Yeah.
So there's no direction in terms of the kind of conversation that you have.
No, so she had some prompts that if she didn't say anything by, I believe it was 45 seconds into just sitting there, she had prompt questions like, oh, are you here for a study? Like, she was pretending to be just a research assistant in the lab. So it was like stuff like, oh, are you here for a study? Oh, cool. I think another one was like, are you from here? Do you go to Nipissing, or do you go to Canada? Or like really standard questions.
Just wonder if the conversation gets initiated with male, is there a different direction that might go to them?
Right. We didn't code for that, but that's a good question. So like and I seem to remember that quite often, I don't have those like raw numbers. We have the numbers, I just don't have them with me. Is how often did the man actually initiate conversation within the first 45 seconds? And I think it was quite rare. Like it was, and the men who did initiate conversation quicker were not like, they were paired men. So these were men who had reported having girlfriends. And the other thing I should have mentioned is these are all heterosexual males. We had to for the purpose of this research question. Okay, so what did we find? Well, we found one interesting interaction here is that, and I'll try to orient you to the main finding here. So the Confederate's affiliative behavior, as rated by the our 2 coders, the Confederate's affiliative behavior was positively related with the extent to which he thought she was interested in him. But this was restricted to the condition in which they got testosterone. So in other words, this male, the way I like to think of it is the male interpreted her affiliative behavior as she is interested in me as a long-term or a short-term mating partner, but only if they got testosterone. If they got placebo, there was absolutely no relationship between her affiliative behavior and his perception of her interest in him. Okay. The other thing that my student was interested in looking at was the extent to which the male's perception of his own attractiveness. So he asked them, how attractive do you think you are to other women? And they rated themselves on a 1 to 10 scale. And so he added that as an additional exploratory analysis to see whether one's perception of their own attractiveness would moderate these effects. And it turns out that it did. So we have a 3-way interaction. So this is for men who are low self-perceived attractiveness. There's nothing interesting really going on. For guys who are high on self-perceived attractiveness, Confederates' affiliative behavior is highly correlated with the perception of his perception of her interest, but only after testosterone, not after placebo. So this was Stefan's work. Because we recruited these people after an initial study, one of the concerns was that we're going to have a selection bias, and some people want to do this extra study are somehow different from the people who chose not to do it. And we had a whole bunch of personality indices and demographic variables, and we didn't find any difference. So pretty certain it's not a selection bias. We got lucky that we had a similar amount who got testosterone versus placebo for this study. Okay, so then this naturally takes us to asking whether there are sex differences or similarities in the effects of testosterone on some of these behavioral and psychological outcomes. So one of the problems that I've encountered over the years is that the only single dose testosterone administration studies that are out there are based on Van Honk and colleagues. In this case, Tooten was the first author, but Van Honk is the primary, or is the senior PI, where they do sublingual testosterone administration in women and look at its effects on physiology and behavior. So they publish papers all in very, very high impact factor journals like Science, Nature, et cetera, because they find these really robust, big effects in women after single dose. The problem that I have with it is that the increases in testosterone, so this is a normal physiological range in women, and this is that increase you see is 2,300% above baseline. So they push testosterone levels to super physiological concentration. So still interesting findings that they get with their behavioral effects, but the extent to which this applies to normal variability in testosterone is questionable. And so one thing that we wanted to do is to develop a paradigm that yields similar increases in testosterone amongst men and women. And this is just showing you our effects here in men. And we wanted to show that this is 60% above baseline. We wanted to produce a proportionally similar increase in testosterone amongst women. Yeah. So Francesca, my postdoc at the time, did a study where we recruited 20 participants, 50% women, so 10 women, 10 men. We did a within subject crossover design, and we administered the same 11 milligram dose for men intranasal, and we used 0.3 milligrams for women. And why 0.3? It was in part a guess, but it was in part based on previous work that had used a 0.9 milligram dose. It produced like a 400% above baseline increase. So we divided that by 3 to get to 0.3 in hopes of getting about a 60 to 70% increase. And so here's what we find. So for women, you have one woman here that their testosterone levels go really high, so 3 nanograms per mil. The normal range is about 1, a high range is about 1. But what we do is we get an increase in testosterone among women. The peak's around 30 minutes, but it's already about baseline at 15 minutes and comes back down by 120 minutes. This is men. And so we have a similar response pattern. Arguably, the 120 minutes is a little higher in men than it is in women. And then what we did is we standardized within men and women for the testosterone conditions. So we had their baseline set at 0, standardized within sex. And we find that the curves for males and females are very similar. So we show a proportionally similar increase in testosterone amongst men and women. And so now we have a paradigm that we can use to try to study sex differences and or similarities in the effects of testosterone on some of these behavioral outcomes. So these are brand new data. I think I still have a few minutes left, but these are brand new data. We're not yet done data collection with women. So at Nipissing, as I'm sure it is similar here, we don't have any students on campus during the summertime. So we're gonna have to collect the rest of our data in the fall. But we've collected data on 100 men already, which is our sample size. We targeted 100 men and 100 women. So we're at about 50 women right now. The challenge with women is they have to do 2 things to meet inclusion criteria. This is kind of ethics driven to make sure that we're not giving testosterone to women who might be pregnant because it can have negative impact on fetal development. Although arguably a single dose at this low concentration probably wouldn't be bad, but we're erring on the side of caution. So they have to report being on a highly effective form of contraceptive, and they also have to provide a negative pregnancy test in order to qualify for the study. So those are the two things that make it a little bit more challenging from a recruitment standpoint. So in this study, we wanted to leverage the more strength of the repeated measures design in terms of testing aggression on 2 different occasions. So testing aggression, and we also had a measure of dehumanization, similar to what I discussed earlier. So we had those measures before drug administration, then we gave them a drug, and then 30 minutes later, we tested aggression and dehumanization again. So we're able to look at within-person changes in aggressive behavior as a function of whether you got testosterone or placebo, as a function of whether you're male or female, as well as the personality risk index that I discussed earlier. So these are what we have so far. So our findings in men are very similar to previous work, where testosterone tends to increase aggression. The effect is not statistically significant. In women, there's absolutely no effect of testosterone on aggressive behavior. Here, I'm going to show you the exact same personality risk by drug interaction in men. So we don't get the 3-way interaction. You can see at the bottom, it's like 0.14, but our data collection is not complete with women. So we have another 50 women to collect data. So, but we were able to split by sex, and we find the exact personality risk by drug interaction in men, such that testosterone increases aggressive behavior, but only among men who are dominant, impulsive, and who have high independent self-control. And in women, we have no effect of testosterone, and this does not depend or is not moderated by that personality risk index. So, so that's where we're at in terms of our first study that I'm showing you. We have another study that we just finished on competitive endurance in men and women, but I feel like we're opening up the floodgates to study some of these sex differences and similarities in effects of testosterone and behavioral outcomes now that we have this pharmacological challenge paradigm that seems to work pretty well. And this is the last one I'm going to show you. This actually, I didn't think I added this, but I did. We exploratory analysis. We wanted to see whether there was this is dehumanization. Yeah, so for men, we have again an increase in dehumanization after testosterone relative to placebo. And this is different from our previous dehumanization research, because the previous research, men were rating women, okay, on human and non-human emotions. This one here, men were rating their game partner, who was another male, on human and non-human emotions. And so men engaged in more dehumanization towards their game partner relative to on testosterone relative to placebo. And for women, there's no effect of testosterone on dehumanization. So that leaves me with just one last quick slide on challenges and future directions. And so my answer is up after this year, and so it's time to start writing for the next one. And so some of these challenges and future directions are going to be reflected in that new application. One thing is, and I was just at SBN, Society for Behavioral Neuroendocrinology, and there's a big talk about rapid versus delayed effects of steroid hormones. And so what we're studying primarily here is rapid effects. So testosterone is having an effect within about 30 minutes of drug administration. But there's also well-known genomic type effects that might not materialize right away, but might materialize 2 or 3 hours after administration. So trying to look at the effects of testosterone from a rapid versus delayed perspective, I think is going to be important for this type of work. Jack Van Honk's work always waits 3.5 to 4 hours after administration to measure their behavioral outcomes. And so they have found some really interesting effects in women when they have this time delay. So this is something that I think is going to be important to consider. Another thing is whether this is androgen receptor or estrogen receptor mediated. We had a project in works or in the planning phases where we were going to do an androgen receptor blocker. And the problem with that is I didn't feel comfortable with it because some of the side effects of it include serious liver disease. Even though it was a single dose, I just wasn't comfortable with that. But another way to get at it could be through an aromatase inhibitor, which has fewer side effects, and so we might be able to look at that. Acute versus chronic dosing. So this is all single dose, and it's primarily motivated by some of our earlier correlational work looking at testosterone reactivity patterns in response to competition. And so we're looking at acute effects, but certainly looking at more chronic dosing, I think is going to be important, especially in certain populations, like, for example, men with type 2 diabetes. Well, I'm not sure if you knew this, but about 45% of men with type 2 diabetes are considered hypogonadal, many of which can get treated with testosterone replacement therapy, which might improve some of the symptoms like low sex drive, low energy, body mass index, etc. But it might also come at a cost in terms of behavioral effects like increased aggressive behavior in certain men. So I think looking at chronic dosing versus acute dosing is going to be an interesting, potentially like a CIHR type project with health outcomes. And I also have psychological mechanisms. So I touched upon one of them being a reward-related mechanism, but I think that this is a whole unexplored area of research that needs to get a bit more attention in the future. And that's that, just acknowledging some people that are critically important in all this work that I presented. So some of the postdocs that I've had, Valentina, Sean, who are both at Fraser Valley, which is, I think, nearby here. So they're both there, and Francesca. Brian Bird, who's a graduate of SFU, Triana Ortiz and Tracy Lynn, grad students in my lab, and of course Neil and Tanya, and my supervising MDs, Pierre Bonnet and Bernard Goldfarb. Without them, we don't do these kind of studies. And then, all the funding that helped us gather all these data. So thanks a lot for your attention. I'm happy to take any questions here or online.